Hello dear students, welcome to Biochemistry by Revati. I am Revati and in today's class we will learn the reactions of monosaccharides. Okay, as we have already discussed the monosaccharides in the classification of carbohydrates, haven't we? Okay, now coming to the monosaccharides, they are the simplest form of carbohydrates, right? Means they cannot be further broken down into simpler sugar units, right? So example here we can take for monosaccharides are glucose, fructose and galactose. Okay, so now coming to the reactions of monosaccharides, first we start with mutarotation. Okay, it is one of the most important reactions in the reactions of monosaccharides. Let's get started. Okay, here, why monosaccharides undergo reactions? Because they contain functional groups as aldehydes or ketones, right? They make them reactive to undergo oxidation, reduction, esterification reactions. So, these reactions are essential to identify the sugar and to study the structure of sugars and to release energy in the form of ATP and also to perform most of the biological functions, right? So let's start with the first reaction, that is mutarotation. Okay, mutarotations. What is mutarotation? It means the change in the optical rotation of a sugar solution when it is dissolved in water until it reaches the equilibrium, means a stable balance between the alpha and beta forms. Let's see in detail. Now we have seen in the definition of mutarotation, there will be a change in the optical rotation. Now what is this optical rotation again? It is the ability of certain substances such as optically active compounds, right, to rotate the plane of polarized light when it is passes through their solution. Example, here glucose solution will rotate the plane of polarized light when it is passed through their solution. But if you take in the case of plain water, it will not rotate the plane of polarized light, right? Let's see with the simplified example. Okay, now I have taken two beakers, right? Here one beaker contains glucose solution and this is a plain water, right? When the light is allowed to pass through the solution, means both the glucose solution and the plain water, here the glucose solution will rotate the plane of polarized light, but plain water will not rotate. So this is nothing but the optical rotation, right? Now coming to the structure of glucose. So here glucose mainly consists of two cyclic forms. So those are what? Alpha glucose, beta glucose. Let's see the structures. This is the C1 carbon, okay? So if you see the alpha glucose and beta glucose, there will be a change in configuration of C1 carbon, okay? If the OH group is down, we will call it as alpha glucose, right? Here you can notice that OH group is down. If the OH group is up, then we call it as beta glucose. Now these two forms we call it as anomers, okay? Is it clear? So in water, alpha glucose and beta glucose don't stay separate. They keep on changing to each other through a small open chain form. Means here the alpha glucose will convert into open chain glucose. Again, it will convert into beta glucose. Is it clear? So in the definition, we have seen that there will be a change in the optical rotation. Now, if you take alpha glucose, it will have specific rotation as plus 112 degree, and beta glucose will have specific rotation as plus 19 degree, right? When these are dissolved in water, the rotation don't stay at plus 112 degree or plus 19 degree. It gradually changes to plus 52.7 degree. This is the equilibrium constant value, means until the equilibrium is reached. Okay, so why it is because here the alpha glucose and beta glucose are interconvertible until the equilibrium value is reached, that is plus 52.7 degree. So if you take the flow chart, alpha glucose having the specific rotation as plus 112 degree, okay, and here the beta glucose will have the specific rotation as plus 19 degree. Now through the open chain form, they will reach the equilibrium, means interconversion of alpha and beta glucose will take place. Now here they will reach the equilibrium constant value of how much? Plus 52.7 degree. Okay, is it clear? Now have you understood what is mutarotation? Mutarotation is the change in the optical rotation of a sugar solution when it is dissolved in water until it reaches equilibrium, means a stable balance between the alpha glucose and beta glucose. Okay. Now coming to the importance, mutarotation can shows that glucose can change the forms, alpha and beta forms, right? It proves that sugar can open up into open chain form, right? And it also works, means it also acts as reducing agents, okay? And in our body, enzymes will identify the correct form, whether it is alpha or beta form, right, to work. Even it is important for the metabolism also. So, is it clear? That's all for today's video. I hope you have understood the concept clearly. If you have any doubts regarding the concept, please comment on your doubts, okay? And if you found this video helpful, please like, share and subscribe to Biochemistry by Revati for more simple and clear explanations, okay? And do support me, and thank you for my 10 subscribers. You are giving a lot of support to me, okay, and thank you once again. So in the next video, we will continue with the same reactions of monosaccharides only, because we dealt with mutarotation in this class, okay. Further reactions we will learn in the next class, okay. So until then, keep learning and stay curious. See you in the next class.